nematode deck up too now, so we'll be starting on that shortly. But we will not finish the second deck today, we'll probably just get through the cestodes and tapeworms. Alright, just so we have a good understanding of like how this is done in the lab, a lot of what we're talking about initially are going to be intestinal infections. So understanding how stool and peas are done over in parasite tests is important. We'll spend some time talking about that. But stool specimens are a major specimen sort for doing parasitological testing. Talk about that a little bit. We won't talk about blood much until we get to like the fourth deck. We're talking about blood and tissue parasites. But just a little bit of a primer on that today within thick smears, how those are done, why they're done. Tissue biopsies, especially when we talk about tissue parasites, talk about what types of things we'll find in those specimens. Urine we don't use a lot of, but we do if we're testing for like Schistosoma hematobium, which is a trematode that can cause urinary issues. And then some of these less common specimens, it's kind of everything under the sun, but we do have parasites that can cause meningitis, so sometimes we get CSF samples, we get parasites that cause lung infections like Peridominus, many others as well, especially Staphylococcus and some other pollens. Corneal scrapings if you have keratitis, amoeba species, duodenal aspirates, Giardia and Cryptosporidium. Our one and only true STI, Trichomonas vaginalis, we do general swabs for that one. And then skin scrapings when we start talking about ectoparasites. For stool specimen recovery, anybody ever had to collect a stool specimen from a patient? Okay, like a little bit of experience doing that. If we're talking about parasitology, parasitic diseases, the challenge is those trophozoites. Diagnostically, trophozoites are gold. They are super easy to identify. The cysts are a little bit tougher. So if we can, we like to get a specimen while the tropes are still alive because they start falling apart as soon as they're out of the body, warmth of the body. So a lot of this is kind of geared towards the clock starting after the person passes that stool specimen. You do not have a lot of time to get that specimen down to lab and look for those trophozoites for diagnostic purposes. There's a lot of things that can screw up your ability to find and recover, particularly those eggs in O&P exams, open parasite exams. Some of those things would be things like, wouldn't necessarily think of, but antacids, anti-diarrheal compounds, anti-motility drugs, pyramide, things like that. Mineral oil, all of those things will screw up particularly your ability to find the trophozoites. Maybe not the cysts. And it will also screw up the morphology if you do see them. You really need to have those those structures intact. Antibiotics don't really target anything other than bacteria, right? That's why we're always saying don't use antibiotics unless it's bacteria. So why would we care about whether or not somebody's been on an antibiotic if we're trying to ID parasites in their stool?
Thing you're killing with antibiotic might be feeding the parasite.
More likely than not, it's also messing them up. So think about an antibiotic like clindamycin, kind of a nasty antibiotic, a lot of adverse effects. If it's causing you as that giant eukaryotic 70-kilo host side effects, think what it's doing to that poor worm that's living inside of you. Okay? I mean, it's not specific. It's not like we can use clindamycin to treat a tapeworm infection, but it's probably not good for it. It'll probably screw up its egg production. It'll have effects. So just knowing that is important for diagnostic purposes. If at all possible, it's better to get that stool specimen from that person who has diarrhea, intestinal symptoms that you suspect might be parasitic, before they go on antibiotics. If not, whatever, you deal with it. But it is preferable to get that stool specimen fresh and before any of this other stuff. Barium sulfate, that's used for doing contrast imaging. You do barium enemas in patients where they have to really get their intestines clean as a whistle and then we get this on. But that will really screw up your ability to recover parasites for a long time, like a week or two. It might even clear up the ER infection. I mean, yeah. So knowing that is also important, labeling those specimens properly. Anything that will form crystals can mess up your parasite ID. If you do get a stool specimen, so we used to execute multiple surveillance protocols for diarrheal surveillance, and we would, Americans, Romanians, Thai, Kenyans, Georgians, like a lot of different populations for these surveillance studies. Where if you're diarrheal, we give you, or you have diarrheal symptoms, we'll give you what we call a cowboy hat. If you've ever seen them, it's a bit descriptive, right? It's these large, wide-brim collection containers that you give somebody so to prevent them having to struggle with like aiming issues, splashing issues, all sorts of things. You just want this stool specimen to be pristine when it gets to the lab, right? So cowboy hat, and then after they deposit the specimen, then they can transfer it to a nice, tidy little container for you. But to start off with, a nice big container like that that's clean, that's not that will contaminate the results. Again, like a little splash of water or urine into that specimen can destroy or damage those precious little tropes we're trying to ID. So all those things are important. I know a couple of you have participated in those surveillance studies, and been read in on how to collect stool specimens, but fun stuff. And the reasons are basically that you're trying to preserve your ability to recover as many organisms as you can. In terms of the O&P exams themselves, as we start talking about helminth infections and intestinal protozoan infections, with the helminths, like the ones we're talking about today, the tapeworms, they're egg-producing machines. They shed tons of eggs kind of continuously in stool. So because of that, we don't usually have a hard time finding eggs in specimens from people with tapeworms. If they have a intestinal protozoal infection like Giardia or Cryptosporidium or Antinia histolytica, those are intermittently shed into stool. So that means you can get a stool specimen from somebody who legit has diarrhea and find nothing in there, even though it's hard to find in the first place and you might have missed it. So because of that reason, the intermittent shedding from protozoa, we like to collect stool specimens on multiple days, for at least 3 days before we even consider calling that negative. I mean, it's just a human being looking at a microscope, subjectively trying to find these things. So it's not the most sensitive way to test it in the world, but we do miss quite a few of these protozoal infections if we're trying to do it with microscopy. It's important to get rid of the fresh specimen if there's a way to get that to lab within 30 minutes of being passed by that person. If not, then you can add preservatives, but remember, fresh is best when it comes to stool. Representative amount of material is usually considered to be about the right amount for the microscopist to work with, or the lab to work with. And again, if we do report out no parasites seen, that's all it's saying. It's not saying negative, that's too strongly worded. It's just saying we didn't find anything. Yeah?
And for the preservatives, is it that don't add it to the specimen, or is it that the person should do preservatives?
So if you get a fresh specimen, if you can get an aliquot without any preservatives, so they can do like a wet prep, add some iodine or some saline, do that, but an aliquot is just a portion of it. And then the rest of that you can add preservatives to preserve the cysts and tropes and other parasitic structures are in there. Yeah. To drill down on this age of specimen, that's what we were just talking about. That relates to consistency of the stool itself. We have what's called the Bristol scale. You've heard of that before. It's a 7-metric scale that grades the consistency of stool from 1, which is like basically passing hard nuts, right, like super hard stool, all the way down to 7, which is super watery, like looks like it's electrolyte solution, very watery. Your ability to recover trophozoites diminishes as stool gets harder and harder. So if you want to collect trophozoites, you will have the highest probability of collecting them from watering stool. You might be able to see some in loose stool, maybe even soft stool, but by the time you get up to form stool, the trophozoites have either gone into cysts or they've been destroyed. So you won't see any of those. In terms of timing, within 30 minutes, that's that limit that you have in order to get those trophozoites under a microscope. watery stools. The soft stools, because you have a lower chance of finding them in the first place, up to an hour. And then if it's formed, you're probably not gonna find any trophozoites at all. So because of that, just stick it in the fridge, preserve it, whatever, but you're not gonna find any trophozoites. With preservatives, 10% formalin and PVA, polyvinyl alcohol, are the two most common preservatives we use. We have slightly different purposes for using formalin. That's better for cysts and then helmintic eggs. PVA will preserve the tropes if they're there. When you get a stool specimen from a patient, I'm talking in terms of the lab, but it's good for you guys to hear this too, because you're the ones who are probably going to be seeing the diarrhea patients. So when the lab receives that specimen from the patient, we do a gross examination of the stool specimen. We look to see whether or not there's visible blood in there, whether there's any mucus in there, which would be suggestive that the patient has some inflammatory process going on. You might see worms themselves, like worms passed in the stool. Sometimes you see, it's described as like moving grains of rice. Those are the perglotids from tapeworms, teeny tiny little pieces of rice. And then we note the consistency on that Bristol scale that I was talking about before, and then we proceed with this microscopic examination of the stool specimen. With the microscopic exam, from the fresh stool without preservatives, if we can, we'll add some saline, add some iodine, throw it on a slide, put a cover slip on, throw it on a slide, and see if we can find those tropes. If they're still alive, we might actually get some motility as well, motility information, like see if they're flagellated or if they're ciliated or whatever. We can also count to see if there are erythrocytes there, if there are leukocytes in that blood specimen, other information that would be of diagnostic value. Some of those protozoan infections can be scamped in stool specimens. And because of that reason, if we can concentrate those parasites, we have a couple of techniques to concentrate the parasites based on the density. Parasites are less dense than zinc sulfate, so they'll float to the top, that's one concentration. But they're more dense than formalin, so they will settle to the bottom of a 10% formalin solution once you shake it up. Then you can also remove some of the debris that's in stool and get much better specimen. And then there are a bunch of different stains we use. You don't have to know this chart, I put it in here. So like once we start talking about doing a trichrome stain for some like Giardia, or an acid-fast stain that we only talked about with Mycobacterium, but is also useful for some parasites like Cyclospora or Dyspardia, so that you have some sort of reference to look back at and how that's done. Okay. But just know that with those stool specimens, we may have to concentrate them, and we oftentimes use common stains to look for those cysts and tropes. Another test we do for stool, the field of parasitology is sort of robbed from other fields, is fecal occult blood test. I might have mentioned it when we were talking about bacterial dysentery. Sometimes you see gross blood in a stool specimen, and sometimes you don't. So if you don't, and you suspect they might have a little bit of dysentery, a little bit of blood, you can do this fecal occult blood test, which is a really sensitive way of picking up whether or not there's some blood that's gone into the stool. It's actually more useful for diagnosing things like colorectal cancer, ulcers, other things. But if you have amoebic dysentery due to Atypical silica or some damage from a tapeworm or hookworm, this will pick that up as well. It's pretty sensitive. Okay, so that's all we're going to say about stool specimens for blood. Thin and thick smears. If you suspect your patient has malaria, babesiosis, leishmaniasis, other infections like that, you can do thin and thick smears on that patient's blood specimen. Anybody ever done a thin thick smear before? No? Okay, a little bit. So with a thin and thick smear, you take that blood specimen, you drop a drop of blood on a slide, just in like one concentrated drop, and you let that completely dry. If you were to look at that, just that would give you a look at the most concentrated look at those blood cells to see if there's evidence of parasitism on that base. For the thin smear, you take another drop of blood and you put it on that slide, and then you take a spreading slide and just at a 45-degree angle whisk it across to spread that blood specimen out. you stain it with something like a Giemsa stain. So this is unstained and this is a Giemsa stain, thin smear at the bottom. And what that'll do is like spread that parasite out so you can look at it under high magnification and get morphological details to the point where you can actually speciate whatever type of plasmodium that person has. Is it falciparum, is it valley, is it vivax? So that's what a thin smear is used for speciation and a lot of detail. Kind of moving all over the place back to the lower GI tract of pinworm prep. It's a famous test, tested for the presence of pinworms, which are it's a very common infection. And as you can see in this picture, I was thinking about making this bigger, but I think you get the point. This is an infected person who has adult worms crawling out of them. And it causes intense itching. That's basically the worst of the symptoms. You get a lot of itching in that paraanal region. For diagnosis, a lot of times, this is more common in kids because of their habits, whatever. But with kids, it's usually diagnosed on the basis that they have this uncontrolled itching, particularly at nighttime when these worms are crawling out of their bodies. So the female worms will live in the lower part of the GI tract, the lower part of the colon. And then at nighttime when the host is resting and still, it's a good time to crawl out, lay some eggs, which can be picked up in the wind and disseminated broadly throughout the entire household, right? So in the morning, if you want to catch highest probability of catching these guys, you take a little piece of tape, something with an adhesive, and you press it onto the perianal area. That's called paddle test or Scotch tape test. And then you submit it to the lab, and they can see if they can find the eggs themselves. Pinworm eggs are super distinctive, easy to diagnose, easy to identify, and there are usually lots of them. And then you can identify the adults too, which are also distinctive. So this is still done. It's not, like I said, it's usually not done very often anymore because it's easy to diagnose pinworm infections with intense itching alone, but that's how you would do it. Don't use cloudy tape because it obscures your ability to identify those tiny, very translucent hyaline pinworm eggs. So, fun test. PCR, thank God I have PCR, right? Love PCR. So clean. So, this is a GI panel for the film array. We've talked about, I think, every single one of these bacterial etiologies, except for Plasmodium Shigelloides. We haven't talked about that one. But we've talked about all these viruses, Sacro and Astro, not a lot of detail of it. For parasites, we're going to talk about these three, Cryptosporidium, Cytospora, and Entomolytica. Those are targets on that PCR panel. There are other companies that also include, parasites on their panels. So PCR is always nice, usually very sensitive, very specific, but it is expensive. You probably have to have a pretty good cause for ordering a test like this. We have immunoassays. Probably the most commonly used one is Vinex Now rapid diagnostic test for Plasmodium species. We'll talk a little bit more about this when we talk about malaria, but this will test for various species of Plasmodium. You can see like PF, PV, whatever, PM. Those are different species of Plasmodium. However, that test has varying degrees of specificity and sensitivity. So it's very good at picking up Plasmodium falciparum, the most concerning one, not so good at some of these other species. Now, that's just an example, but it is nice to have those. They are inexpensive, they are rapid, and when they're available for that particular parasite, they can be very useful for diagnosis. Last couple slides just talking about how parasitic diseases are controlled nowadays. 100 years ago, 150 years ago, 200 years ago, we did not have this wonderful thing that is modern sewage systems. That has made life a lot safer for us in terms of contracting parasitic diseases. And I think this is something we take for granted, what that allows us to do. But even today, we're going to be talking about parasites that have co-evolved with us because we haven't been able to, historically, keep our contaminated fecal matter away from foraging animals like pigs, for example, away from ourselves. So very important. In the US, we have wonderful sewage treatment plants. A lot of countries don't. And they're kind of stuck with some of these parasitic illnesses that are kind of unheard of in the US. We don't get them anymore, but we used to. We used to have a lot of this stuff here in the US before sewage treatment plant facilities. We also know better than to do things like using night soil, fertilizing our crops. What is night soil? Human feces. It's very economical to do that, right? And it's also environmentally friendly. But the problem with using night soil to fertilize crops is you're concentrating human-specific parasites on crops that will eventually be used for human consumption. So if you're like growing up your raspberries or blackberries or cilantro or whatever, you're using night soil to do that. It's concentrating human parasites into that area. Now we know not to do that. Use cells as fertilizer. Water purification. We use chlorine in swimming pools. We use iodine if we're out in the field and we need to drink water to kill parasites at a certain concentration. Some of these parasites are weed bit resistant to things like chlorine and iodine, so we have to be careful about that. But we do have measures to reduce the transmission risk for some of those waterborne parasites. Making sure our groundwater isn't contaminated from leaking latrines, flooding. That was a kind of what happened at Milwaukee outbreak of cryptosporidium back in '93 that I was mentioning. They had sewage treatment or sewage leaking into some water facility, municipal water supply. Filtration systems, if you have them, they need to be replaced and maintained adequately. When we're talking about tapeworm infections today, food safety is important. Big thing, if you're going to be eating something, just make sure it's cooked all the way so you can destroy any larvae, parasitic larvae, or eggs. Environmental controls, pretty self-explanatory, but the housing we have housing standards, we have building codes. A lot of places don't, and they become breeding grounds. We have a lot of information about how vectors, like what types of environments they like to live in, so you can adjust appropriately, control things like thatched roofs. With vector control, meaning like the arthropods, we have some really aggressive methods of reducing those populations, but also just simple things like bed nets when you sleep, mosquito nets when you sleep, insect repellents, wearing protective clothing, maybe not going out in the evening when during peak hours for certain types of mosquitoes. And then like I said, very aggressive methods like using large-scale insecticides to destroy insect populations, sometimes with disastrous environmental consequences. Molluscicides, I bet that trematodes all require that snail intermediate host. So to control those, especially in endemic areas like in sub-Saharan Africa, they'll use molluscicides, like large-scale molluscicides to control snail populations. Genetic control of vectors, a sterile insect technique. We have genetically modified mosquitoes that can be released. We've been working on this technology for 20 or 30 years now, trying to make it so basically the female mosquitoes are infertile, they can't reproduce in a way that doesn't completely destroy or eradicate them. So a couple states in the US are licensed to do that. Florida and Texas. Texas is one of those states that's licensed to release genetically modified mosquitoes. And there are countries that do that as well. Cayman Islands does it, and a couple other countries. And then for protection, you have prophylaxis, like we have multiple prophylactic drugs for malaria. That's a good way of controlling against malaria in endemic countries. Most of you guys, if you've deployed or gone to an endemic area, have been on malarial prophylactic drugs. Immunization, there's not a lot of parasites that we have vaccines against. I can't think of any off the top of my head, but if they're available. And then lastly, for anti-parasitic drugs in general, because these are eukaryotes, we kind of run into some of the same issues that we talked about with other microbes. It's hard to target them in a way that doesn't also hurt us, so we have a pretty limited arsenal. Because malaria is such a big problem, a lot of our anti-parasitics do target malaria or Ritalins, artemisinin, those types of drugs. We're starting to see some resistance to those sorts of drugs, so that's becoming a problem. Even the combination therapy for malaria is becoming problematic. In terms of the drugs that we use for helminths and for protozoal infections, this is the only slide, one of the only slides where I have any information about parasitics. I've taken most of that out of the deck because you will get that in other courses, but just very broadly, we can use drugs like azaquantin, or that's a common drug to be used for tapeworm infections. Albendazole, those types of drugs we use for helminthic infections. Metronidazole, that might ring a bell for you guys from block 2 when we were studying antibiotics. Anybody remember what metronidazole targets? Not a soul. Yeah, exactly. Generation of free radicals during replication. So that is a good antiparasitic drug and targets some of these protozoan parasites like Giardia, Trichomonas, Candida. And then lastly, I'll skip hand washing. That is super important, and don't skip hand washing, but not much to say there. It prevents fecal oral transmission. But education is really important. With these populations, generating infographics like the ones on this slide that tell people with like malaria, the one on the bottom, for example, tells people what clothing to wear, what activities to maybe taper off from doing in the evening hours, how to protect your house, how to get rid of still water, just telling people who live in endemic areas how to control mosquito populations. And in the top there, with cryptosporidiosis, an infographic that the CDC put out to educate people about the likely routes they could acquire crypto. 35% of people acquiring it from pools, chlorinated swimming pools, where unfortunately you've had a sick child with diarrhea in that pool, and that just, everyone swimming in that pool can get sick. It's contributes to a lot of cases and outbreaks of cryptosporidiosis, but also to a lesser extent things like interacting with cattle. If you're talking about zoonotic species of Cryptosporidium parvum, for example. And then children, childcare, daycares where kids are packed together closely, that's been associated with cryptosporidiosis outbreaks as well. So as long as people are familiar with how these things are transmitted, we can really do a lot to bring down incidence of these parasitic diseases. That is it for this deck. Any questions on the intro to parasite stuff? All right. How long have I been lecturing? 30 minutes. Okay. All right, I'm going to put you guys on a break while I move up the next lecture. Before you go, I want to show you these YouTube videos as a primer for tapeworm infections, and then you can go on break with these happy thoughts, okay? So sometimes it's just better to see things rather than describe them if it's possible. Videos. So we're going to be starting to talk about tapeworms, right? And by and large, there's not a lot of symptoms associated with tapeworm infections. Oftentimes, they're discovered accidentally when people do colonoscopies. So this is an example of a beef tapeworm. Scientific name is Tinea sauginatum. It was discovered during a colonoscopy. Just want you to see how long it is, how it moves. Maybe this is not super high resolution, but maybe you can see that it's segmented, and that those individual segments are called prolatids. So imagine just being, you're a colonoscopist, and you stumble across something like this unexpectedly. That would be quite a surprise, and I'm sure the patient was probably unaware as well. That's a very clean colon too. You did a good job. A+. Good job.
Can you snatch it out of there? Or you gotta do something?
What's that?
Can I snatch it out?
Oh, I don't know. I don't think they would do that. I think they would probably treat with an antiparasitic. But I do have a link for when we talk about Tinea solim, the pork tapeworm, that shows them pulling out a 6-meter tapeworm from from the mouth. So gums anesthetized, I don't know how they did it, but I mean, the picture shows them pulling out this tapeworm from the oral cavity. It's insane. This is a fish tapeworm in a human colon. So interesting, like it's bile stains. Let's rewatch that. That's why it's yellow like that. And it has a very wide body relative to cutaneous. It's also, look at the colon itself though, like you see all those blood vessels in the colon? That's an irritated colon. So there might be a little bit of pathology with that patient. I mean, they probably are experiencing some uncomfortable symptoms at that point. And then lastly, I'm sorry, and then I'll let you guys go about your brain. But this is in Australia. Oops, sorry. She's saying some kind of interesting things. So this is where they say the term called environment trap that they pulled out of a cat, which is a definitive host for this tapeworm that humans can also be infected. So small little cat, and they pulled out this 2.7 meter tapeworm, which is over 8 feet. That's a whole lot of blood. And each one of those segments is like it's a egg-producing machine. So it just produces eggs continuously, month after month, year after year, long-term infections. The definitely macroscopic, like you see handling this tapeworm. Some of them are much bigger than that, but that's just pulled out of a little cat. The entire worm. Probably got that. Anyway, so that's all. I just wanted to front load those. You guys can go on break now, and then we'll start the session then.
So the coffee itself is I drink espresso morning, so that is fun. The other one, yes. Very popular.
Yeah, I would never. Yeah.
Yeah. Well, mainly because I am American and that retro 60s look. I was talking so hard. I'm like, I was in there. Yeah, for real. Like the look was it was like yeah. It's like the yeah. Like I know a lot of people didn't like I was like how the big was how the rush and go like too much into it. Like kind of like sharing that kind of like a I didn't mind it. I didn't mind it. I thought the overall the movie was great. Overall the movie was great. The overall look like yeah. I didn't appreciate the CGI.
Okay, you guys ready? There you go, thank you. Alright, so talking about the tapeworms today. As you saw in that video, these are very long, flat, ribbon-like organisms. Do have variable length, some of them to be very long. That 10-meter tapeworm is in the center of the picture there. That's the distance from me to Jake. That's a very long tapeworm. It's so animal's gut. That's what starts off. In gestation, it is oftentimes when people all around, you're the definitive host. The eggs will hatch into larvae. Larvae have these little skulls and hooks in them. So when they get down to, they'll access to the stomach, and then the larvae will, with those little hooks that they have, will latch onto the intestinal epithelial layer. Once they latch on, they start developing. So they start off as a tiny little larval worm, and they start maturing into adulthood in the intestines there. They start producing these sections that we talked about called proglottids, really tongue. And they start from the scolex, there's a little quote unquote neck there. Just keep kind of growing out, so the longer and older that tapeworm is, the more proglottids it accumulates. It gets to be quite lengthy over time. You can have these infections for decades. It's usually just you and one tapeworm. There's not like a lot of tapeworms you get. It's like you and your tapeworm together for 25 years. So it's a significant relationship that you have there. With this head, which is basically just an attachment structure, it stays attached to the intestines your tapeworms tracking that you have breakfast taco coming, and they can latch on and like move a little bit, but they need to latch back on someplace else when they get that food supply. In terms of eating, that scolex is a head only in name, brain, and there's really no mouth there. They acquire nutrients through the entire body. It just absorbs nutrients through the complete digestive system. So they're just absorbing whatever content's but yeah.
If the tapeworm breaks, do you have two tapeworms or?
Oh, you mean like the ability to regenerate and latch on and want to be tapeworms? No, that scolex is a scolex of worms. So you're going to regenerate a scolex. If they lose that scolex, that life cycle is probably terminated. That probably will be tapeworm. But they do kind of continue to grow until they have natural death. Their lifespan, depending on the species, can be 25 years or so. So they'll continue to grow. I showed you that picture of a grizzly bear or brown bear that had a very long tapeworm hanging out of its body. In nature, you can see organisms like that. I've never seen that with a human, but you can occasionally see the worm itself coming out of people. It's not very common though. Usually what does happen is, as I said, you kind of continue to shed eggs into the feces, or sometimes you see your blotted come out. Anybody, you guys have pets, right? Anybody ever see a tapeworm come out of a dog or cat? When I adopted my cat last summer, April 2024, got it from a very crowded boarding situation. He had a flea tapeworm, and I watched him just deposit a little percolated right on my tongue as I was working on a lecture. Wow. So I got my phone and I videotaped it. I'll bring it up tomorrow. It's a really cool little video though, but it's just like, I was like, oh, 30% more horrified and, 70% amazed and appreciative. This cool tapeworm that's coming out of it. Yeah, 37. But yeah, and then I took that stuff, I took it in a little bag, took it over to his bed, and I was like, I get some processed for my cat, and they insisted on doing O&P. I know exactly what this is. They charged me $80, whatever, for the diagnostics. But yeah, I mean that's common. You see these little a lot of sometimes coming out. It scares people. There's a psychological effect. But in terms of the actual pathology of the person, not that much. We'll talk about that. But the classic human endemic tapeworm isn't doing a whole lot of damage because it's the definitive host. Because we learned a huge amount of damage through definitive host. Okay, so that whole body is called a strobila. Depending on the life cycle, there may be multiple intermediate hosts as we go through all these different examples of tapeworms. Humans ourselves can be intermediate hosts once we talk about Echinococcus species, granulosus, and multifocularis. You'll see that when we serve as intermediate hosts, we tend to get a lot of damage. So those are life-threatening diseases, really nasty. And yeah, they reside in the intestines, so they're not going to migrate out into your brain or anything crazy like that. They can stay in your intestines happily, siphoning off calories and sharing the brain. The first two organisms we're talking about, Taenius solium and Taenius saginata, the head itself, that scolex, is not a diagnostic value because it's going to stay stuck up in the intestines, so you're not going to see that. But if you were to see it, you would see, in the case of Taenius solium, these hooks called rostellum, and then these suckers around the edge. Taenius saginata has no suckers, no rostellum, and Taenius solium has rostellum in the center and the suckers. Most tapeworms have those suckers so they can latch on, help them latch on. Methylocobalamin has grooves instead of suckers, but that's some sort of attachment structure like that. If you are lucky enough to get an entire proglotid and a stool specimen, that's money for diagnostic purposes. It's very easy to identify most tapeworm species proglotids. Here we see a couple that have, I think, a carbonate counterstain, but the same thing we were talking about for Cryptococcus, they just inject that into the genital pore there, and it will stain the uterine branches of that proglotid. And then for diagnostic purposes, so count the number of uterine branches in that proglotid. If it's Taenius saginata, it's usually like containing psyllium. You don't have to know that, just know that that's how we identify based on the blood. And then the eggs themselves can be useful. So we'll talk about eggs, which are far more likely to see specimens in OPs. So not always, but sometimes useful. For the subsets that we're talking about in this lecture, things here, this is all about ecology. It's understanding the relationship between humans and these different animals that we have transcycles with. Sometimes we are accidental hosts, like with Spirometromycete, we're kind of getting in the way of a normal cycle between predator and prey. We don't eat these copepods and cold-fronted vertebrates normally, but if we do, we can acquire a disease that we're not really adapted to be definitive hosts for. Same with the Echinococcus species, granulosis and multilocularis. So each of these warrants its own individual studies. We can learn how it's transmitted and how it's diagnosed and what the presentation is. But we'll start off with kind of the easy bread and butter tapeworms, the pork tapeworm and burn tapeworm. Okay, with the pork tapeworm, your screen has both life cycles on it. We're going to talk about the one on the left first because we have a twofer for this organism. This is a fairly common way of contracting teniasis and teniasis. It is basically, it's not fecal oral route. What's happening here is the route of entry is to the mouth. You are ingesting it, but what you're ingesting is mealy meat with larvae in it. So the meat right there, you hopefully have never seen meat like this in your whole life. If you have, hopefully you didn't eat it. If you did, hopefully it was super well cooked. But this is how it's attracting disease. If you eat this mealy meat that's undercooked, those have larvae called sister cerci. That's the plural circus. Those larvae will persist, meaning they lose that coating in your small intestine, and by the time they get into your small intestine, they have that scolic and then attach to your intestinal epithelial wall and grow into an adult. This takes usually a couple months to mature into adulthood. And it takes a couple months before they're sexually mature. So they're hermaphroditic, so these spermatozoids will become able to self-fertilize. It has male and male parts. So they start self-fertilizing, and then you get eggs released into the stool. And then you continue to get eggs released into the stool for as long as you have that tapeworm. So you can be infectious, but usually not self-infectious. As you release these eggs, if you are not going to the bathroom in a normal toilet that you can flush and have it disappear forever. If you're out the bathroom, let's say in the countryside, and just defecating out in the field where pigs are foraging, what happens is pigs forage, they consume whatever is contaminated with your feces that has these eggs in it that are tough, right? They can survive being out in the environment like that. And the pigs contract this disease and become the intermediate hosts. Those eggs will develop into oncospheres, which are basically another larval form. It'll penetrate the intestinal wall of the pig, and then it'll develop to that sister circus in the pig. That causes some pathology of the pig itself. They'll get muscle soreness. That usually insists in the muscle tissue of the pig. That's what you're eating when you eat that pig. Yeah.
What was the word you used to describe the meat?
Mealy, like mealy apple, rotten, kind of. This used to be super common for thousands of years. We didn't have a lot of ways to control consuming larva-ridden mealy meat, but we do now. USDA comes through, inspects meat physically still. They'll go to slaughterhouses and examine vascular muscles and other indicators whether or not you have contaminated meat. And they'll check to see if any of those carcasses have evidence of having sister side. Okay, so that's one presentation. Remember, that's ingesting mealy meat. The other presentation is fecal oral route. Fecal contamination. You're ingesting the eggs themselves. So here you're ingesting the larvae, and here you're ingesting the eggs. Different infected stage, different pathology. You have to know that for the exam. Very, very important. The pathology here is once you ingest those eggs, you basically are the pig or the intermediate host in this scenario. Okay, so this is a human ingesting eggs from another human, and basically, like the same thing happens with what happens to the pig. Those oncospheres hatch, they get our intestinal wall, go into circulation, and as they go into circulation, they're probably going to land, statistically speaking, because a lot of our body weight is muscle, they'll probably go into muscle tissue. And there, they will set up shop long term. What their strategy is to be eaten. So they're there for the long haul. They can be there for years, consistent in your muscle. The only signs and symptoms if you have muscle cysticercosis is you get a little bit of soreness, usually. So maybe your bicep hurts a little bit, or it goes consistent with the arm. Usually not a big deal. And eventually those larvae, if you're not eaten by some carnivore, which you probably aren't because you're an apex predator, right? Then eventually those larvae will die, and you will get an immune response to those larvae. Even that's not that big a deal, but symptoms do worsen once those larvae die. Other bigger problems are when those larvae don't go into muscle, but when they go into vital organs like the lungs, sometimes they can go up to the eyes, and sometimes they can go into the brain. Unfortunately for the brain is one of the more common sites where these larvae go. And then you get something called neurocysticercosis, NCC. In terms of infectious causes of brain infections, this is up there. It's probably underdiagnosed, but you see this on MRIs sometimes, these calcified larvae in the brain, like that. So, there's a very well-known example of a celebrity who has NCC, and I want to say his name, but I can't. Some of you might know this individual, but there you go, Sean. Yeah, the latter. But the symptoms were, I mean, if you were to write like a case report, it would be a 52-year-old male, I think he was about 52 at the time, who had a travel history to parts of the globe that are poor hygiene, and then he developed signs and symptoms of neurological issues like brain fogginess, confusion. Yeah. So, pretty common. This kind of summarizes everything we just talked about. Going back to the intestinal tendinitis, where you're eating really meat, this tends to be in places where they don't have really good meat inspection programs and where feces are released into areas where pigs are foraging. So eating undercooked or raw pork, most of those symptoms as that adult tapeworm grows are going to be very mild. You might not know it at all. Might be completely asymptomatic. If you do have symptoms, it will be things like diarrhea and a little abdominal pain. For bone stems, you find the proglottis cells, or you can find the eggs. The reason why the eggs are a little bit less reliable for Taenia solium is because they are identical to the beef tapeworm eggs, just like Taenia saginata. There's a link to this guy up here. You can see that this is what I was talking about, a case report in India where they found a pork tapeworm and they extracted it. For whatever reason, they made the decision to extract it through his mouth rather than treating with drugs. But they extracted it and they laid it out like a trophy. Yeah.
Is this not trichinosis that is the same as trichinosis?
It's not, so totally different. Trichinosis, that is a nematode infection, but it's the pork. It's also associated with pork as well. Trichinella spiralis is a pork tapeworm, or a pork nematode. Okay, so that's intestinal teniasis. Make sure you don't get that confused with the other form, the cysticercosis, which would be either the muscle, the eye, the lungs, or the brain. That's the most concerning one. That is fecal oral transmission, and the infective stage is different. It's the eggs, not the cysticerci, not the larvae, the eggs. Moving on to the beef tapeworm. This is much easier after talking through Taenia solium because there's only one presentation of beef tapeworm. Here you also ingest mealy meat, but this source is from the cow. It's evolved to use this as an intermediate host, so only the cow. It doesn't use pigs, it doesn't use other intermediate hosts. There's a picture of a beef carcass that also has a bunch of larvae embedded in the meat. We ingest it and the same thing happens. Larvae matures to adulthood in our small intestine, so that's for a very long time, not a big problem, and then we pass to proglottids and eggs. Here's a picture of an egg where it looks like a bioman, yellow, kind of bile-stained egg there. It looks identical to Taenia solium eggs. Can't be used for differentiating them from Taenia solium. Usually that doesn't matter for treatment purposes, except because of the cysticercosis, you might have to treat with additional drugs, whereas with this, it's very simple praziquantel. A couple pictures of this tapeworm down here. You can see the segments, you can see where it's latched onto, but pretty straightforward. Any questions on the tapeworm? Yeah. The proglottids are easy. You can tell if you get a proglottid, you can count those uterine branches, and the shape is different, the morphology is different, and you can diagnose from the proglottids. Yeah. And also, if maybe a little bit of patient history too. If that patient doesn't eat pork, it's probably beef tapeworm. There probably is some PCRs out there available that might be hard to come by. Or not, it wouldn't be a routine thing.
And so this is both larva or egg, or
No, this is larvae only.
I mean,
Yeah. So here's a question for you guys, and you kind of stumbled across a good question. With Denisonium, it is larvae or egg, right? If equal as possible. Why couldn't this egg cause an auto-infection if you were to come across it in oral product? Any ideas? It's because it has evolved to require certain environmental stimuli that you can only get in the digestive tract of a cow. What do you guys know about the GI tracts of cows? They've got like 4 stomach compartments, right? They've got the rumen and the whatever. So each one of those stomachs has a different environmental cue for this larvae to exist and go into circulation. We don't have that complex stomach signaling. So if we were to ingest the eggs themselves, that would probably destroy those eggs. They wouldn't be able to hatch. We don't provide the right environmental cues for that. So because of that, we don't get cysticercosis like we could get with Denisonium. Okay, just one presentation. Okay, diphyllocrisis, fish tapeworm, causes diphyllocrisis. The name itself, Diphyllobothrium, refers to the fact that this thing on the scolex doesn't have those hooks and suckers, it's just two different grooves that it uses to latch onto the intestines. Once we ingest it, it's kind of the same. They are the definitive host, so it matures in our intestines just like before. It starts producing eggs. There's an aquatic theme to this one. You release the eggs need to get into some sort of water source, which is common in parts of the world where this is a more prevalent disease. They get into the water, they mature into a form called portosphidium, which is able to kind of swim out in the water a little bit. And the portospidia, plural, get ingested by something called copepod, which is like a tiny little crustacean, cyclops and other species of that. Then you mature a little bit in the copepods, and the copepods in this food chain will get eaten by some sort of fish, usually freshwater, like a perch or something like that. And then we eat that fish that is undercooked or raw. Okay, and then we acquire the disease that way. In terms of the symptoms, once we acquire this fish tapeworm, they're pretty much the same as when we get tapeworms from beef or pork tapeworms, but there's an unusual thing that makes you vitamin B12 deficiency. This is one of the longest lived tapeworms, so over the years, as it grows to considerable size, producing 1 million eggs a day. I mean, that's insane, right? A lot of eggs. As it's producing that many eggs, it is leaching your body of vitamin B12. So you get some deficiency there. And that's part of the treatment after you put up with praziquantel. Put that person on vitamin B12 supplements for a while. In terms of diagnostics, this is unusual for the egg. It has an operculum, which is basically a lid. You can see that operculated egg right there. When it gets mature enough that operculum opens up and pops off, and the little larvae will come out of that egg. The proglottids, if the person releases proglottids in the stool, the proglottids are distinctive too. You can ID from these. They're very, very long, very pretty sets, middle part. Proglottids. for prevention here, just to make sure you have fish thoroughly, there's a couple associations, like FE associations with dichotherapsis. One is just normal campfire cooking. It's not very common in this part of the world, but if you're cooking fish you just caught on a camping trip and you undercook it, cases of dichotherapsis similar to that. We also have eating sushi and ceviche. Ceviche, what that is like freshwater fish which isn't really cooked, but it's kind of pickled with acid juices, right? So that usually does the job of preventing bacterial growth. It doesn't do anything for preventing parasitic growth. So pickled things in general aren't going to save you from parasitic infections. It's another thing to know. Piscivore. This is a Scandinavian thing. It's basically like a fish ball, minced fish ball. So it's more prevalent in Scandinavian countries. There are cases up in Minnesota and Wisconsin of this, where they have a lot of Scandinavian people. And gefilte fish. Have you heard of gefilte? Yes. Gefilte is like it's a Jewish cuisine. A lot of people eat it during Pesach, but multiple different types of fish mixed and packed in together. But they're cooked. When people usually acquire dichotherapsis with gefilte fish, it's when they're tasting the fish as they're cooking before it's fully cooked. So they acquire the parasite that way. Okay. But the end product should be perfectly safe to eat. You shouldn't taste it while you're cooking it. Okay, Spirometra mansoni, this snake tapeworm. The disease it causes is called sporganosis. It's an individual larvae are called sporgana, plural, or sporganum, singular. This is kind of an interesting one. This is the first example of a parasite where the human is not the definitive host. I guess they're an intermediate host, but we're an accidental host. Remember when we were talking about accidental hosts? We're also a dead-end host in this scenario. So let's talk about that with the lice episode. Normally, this is a parasite that's transmitted between dogs and cats. They eat cold-blooded vertebrates like fish, snakes, and frogs, things like that. So the cats and dogs are the definitive hosts. The worms grow up in their intestines. They will shed those eggs out in water. It's another water aquatic parasitic transmission cycle. In the water, they mature much in the same way that Diphyllobothrium larvae mature. They become Corsidia. Those Corsidia get eaten by Cyclops, those little copepods. And then you have some multiple intermediate hosts that ingest them along the food chain. Where humans come into play is we can basically, interesting in the way that cats and dogs are seeds, we can eat the fish and snakes, and we can eat the frogs and acquire this parasite that way. This is associated with cultures that do that. There's still some places where people eat snakes and frogs and toads and things like that. You can also acquire it through drinking water itself. Those little cyclops or water fleas, copepods, are pretty much microscopic. You can't see them. So if you drink unfiltered water that has copepods and it's infected with this parasite, you can get sporganosis that way. That's one of the more common ways of acquiring it. Third way that you can get it is, in some cultures, they use the skins of frogs and snakes as poultices. So they'll apply it on open wounds, and, with the thought that there's some genocidal stuff in the skin. They'll clear up the bacterial infection most of the time. But the skins are infected with sporgana, and those larvae will migrate out directly into those tissues. If you use that pulse on an area near the eye, the larvae will migrate into the posterior of the eye globe and can set up shots, pretty nasty eye infections. They can also migrate down the optic nerve into the brain and stay in the brain, dead end host. We're not going anywhere at that point, but you get some more serious symptoms if they do migrate along the nerve into the brain like that. For most of these routes, worms get into human hosts. Since we're not a host, we can't support their maturation to adulthood. So what happens is you get a weird presentation. You don't get a normal intestinal tapeworm development. Instead, these worms, these larvae, will flounder around underneath your skin, looking for the right environmental cues, which you find is actually kind of sad if you think about it. So, mean, sort of was, but in terms of pathology, what you get in terms of with pathology is nodules underneath the skin. So you can really see it in this first figure at the top panel that just popped up. Yeah. This gentleman has a bunch of skin nodules all over his torso. If you zoom in a little bit on those nodules, you can see those have, like, in some cases, larvae erupting out of them. And those are larvae, they're not worms, they didn't mature into worms, they're just larvae. You can treat with antiviruses, but most of the time you have to manually excise those larvae out of these nodules, which can be all over your body. So that takes a while. This is diagnostic though. You're not shedding anything in stool, so no one forget about it, you're not the definitive host, but you can see the larvae that you pull out of those skin nodules. In terms of symptoms, itching, I don't know, having worms or larvae coming out of you is probably psychologically damaging. Usually not too bad. It's when eye infections and when they get to the brain when you get more serious infections like that. Okay, geographic distribution of those two papers I just mentioned, diphyllothriasis tends to be associated with like northern climates, freshwater lakes up north, Russia, Canada, Scandinavian countries, Japan. And the spirometric species tend to be more in warmer waters, particularly in Southeast Asia, cases down there, but you can get cases of spironosis due to spirometric species in the United States as well, particularly with cats and dogs. They have normal access to those parasites. Moving along, Chytococcus granulosus. This is the dog tapeworm. The dog is the definitive host. It's evolved to be the definitive host. Other carnivores as well, but with this transmission cycle, it's usually dogs who are tasked with sheep herding activities. Has a couple other names: cystic echinococcus and hydatid disease. Hydatid disease is a little bit older, but you still hear this used sometimes talking about this tapeworm infection. This transmission cycle, this is associated where you have a triad of sheep herding dogs, sheep and people, sheep herders. Those three together, you get human cases of echinococcus. Normally what happens is you have the sheepdogs, the farmers will feed the viscera of sheep to the dogs like a good source of iron, right? The dogs will eat the viscera, and if sheep aren't infected, they will have this big cyst called hydatid cyst that they ingest as they're eating the viscera. The dogs will then become the definitive host, so the larvae will mature to adulthood in the dogs, will then shed eggs and proglodids out into the soil, and then sheep grazing in the soil will pick up those eggs, okay, and then they will become larvae and they'll exist in the sheep and develop back into hydatid cysts. Hydatid cysts are nasty business. For the sheep themselves, it takes years for them to develop. This is a slow-growing cyst. But basically, those cysts have a bunch of tiny larvae inside of them that are nursed. If they stay intact, usually that doesn't cause a lot of problem for this intermediate host, with the sheep or ruminant. But if they rupture, then those protoscopices, the baby larvae, can disseminate all over the body and develop into new hydatid cysts, can be problematic. Or you can get anaphylactic shock because these are filled with nasty, noxious particles and chemicals called hydatid sand. So this causes anaphylaxis when it ruptures. For humans, when we come into the picture, we come into contact with dog feces that's contaminated with this, and then we become the sheep. We become the intermediate host. We become the ones that incubate this hydatid cyst over the course of years. And the symptoms sort of track with where these cysts develop. If they develop in the liver, which is the most common site development, then we get hepatomegaly, we get jaundice symptoms, obstructive jaundice. When we have the portal system obstructive, we get hypertension, portal hypertension. That presents a lot like hepatic cirrhosis. So that's with the liver. If you get incitement in the lungs, you can have cough, chest pain, shortness of breath, and et cetera, et cetera, et cetera. So these symptoms, compression can also occur as these cysts grow. Some are fairly small, but some of them get to be about the size of a grapefruit, and and some of them have been reported to have 2 liters of fluid in there. So if that compresses a vital organ, you can also have pathology due to compression of vital organs. As if you were walking around with a cyst in you, in any one of these vital organs, the main concern and worry is this cyst is going to rupture and release those cortisolyses and cause ammo blockage. That's really a bad scenario. 70% mortality rate if that happens. It's very difficult to treat. In terms of treatment for humans who have hepatitis disease, it's usually detected with studies, MRIs, radiography, things like that. Hard to diagnose. But there's an acronym called PAIR. It should be in your notes if you're interested in reading about it. But it's PAIR, puncture, aspirate, inject, and then re-aspirate or something like that. But basically, you're carefully killing that cyst with formalin and trying to prevent it from rupturing and spilling that hepatitis in all over the body. That's the way that's treated. Pentacoccus monotypicalis is similar in some respects. It also causes a form of pneumococcosis, but there are a couple differences that are important to note for this tapeworm infection. Its normal cycle and its hosts are similar, but this tends to be a little bit more somatic. So these predators are things like coyotes, dogs, foxes, wolves, also cats and dogs. That contributes to it being more widespread. It is more rare than Echinococcus granulosus, so it's less common in humans. But for those predator animals, they usually are feasting on rodents, which are the intermediate reservoirs. Rodents are basically what we become when we accidentally get infected or accidental hosts in this infectious cycle. But this parasite gets transmitted, shed through the definitive host, the carnivore predator, and we ingest it when we come into contact with these predators. The reason why this is called a velar echinococcus in humans is because the cysts themselves develop a little bit differently. They tend to be smaller and easier to disseminate to different parts of the body. So velar, it's not because it goes into the lungs, but it's because it looks like air sacs. It looks like a villi. That's why it has the name of velar echinococcus. It is sometimes misdiagnosed as tumors. Oftentimes in the lungs, it'll get misdiagnosed, or in the liver is hepatic carcinosis. Definitive diagnosis, you probably have to get the actual specimen out and do that through microscopy, but again, you have the risk of puncturing the cyst, dealing with all the problems that come with punctured cyst. Mortality rate is the same as the other form of echinococcus up there. If you do not treat these. And the treatment is much the same. Surgical resection can be done. If it's in the lungs, you might be able to resect the lungs. Antiparasitic treatments will be long-term, antiparasitics for a certain period of time. In terms of prevention, really it's telling people that educating people that there's a risk of transmission with their farm animals, or if they're interacting with these predators, like if they are working with wolves, foxes, coyotes, things like that, telling them that there's a risk of acquiring parasitic diseases accidentally. But when humans get this disease, we are accidental hosts and we are also dead-end hosts, unless something eats us, which is we can transmit at that point, but otherwise considered to be dead-end. All right, last couple of tapeworms. Hymenopus nina causes, this is a tapeworm because it's so tiny, and it causes hymenelopiasis. Worldwide distribution for this tapeworm, it is the most common tapeworm in the world, most common tapeworm infection in the world. And the reason for that is because it has these optional intermediate hosts. It may or may not use beetles and mice. The mice eat the beetles as intermediate hosts, or if a person is infected with Hymenopus, they can auto-infect themselves. So there's multiple ways to get infected, and it becomes a very dangerous infection for that reason. Very dangerous and common. What normally happens here is you pass the eggs to the definitive host. You can maturation period for those eggs, so they don't require weeks and weeks of maturing out in the environment. You can just auto-infect, consume them fecal oil basically, and then get a hyperinfection. So, multiple parasites developing and maturing in your intestines. So some you have hyperinfection are much more severe. If you have a light burden with just a couple worms, maybe one worm, maybe asymptomatic, but heavier burdens, you start getting more severe diarrhea, more severe abdominal pain, potentially anorexia, headaches, neurological problems. Big concern with children. I mentioned that if you're already malnourished, you're already starting to get calories, this can be damaging, especially this particular tapeworm, because of the ability to auto-infect and cause hyperinfections. The reason why this is so common, going back to these intermediate hosts, is these eggs can pass through beetles and they can pass through mice, and those beetles and mice can get into our food supplies, contaminate things like grain stores. So when we eat, like on the shelf products that have been contaminated with beetles, we can contract the disease that way. Very hard to trace. Just important to have good sanitation and good food inspection programs, but that's the reason why it's such a prevalent disease. For diagnostics, the eggs themselves are pretty distinctive. They've got a 6-foot embryo on the sphere of filaments. I put a description in the notes if you want to go back and read through that, but pretty easy to diagnose. This species is Diminuta. Diminuta is actually larger than Nana. The name suggests it's smaller, but it's significantly larger. Hymenopus diminuta, the rat tapeworm. Some similarities to Hymenopus nana, but some differences are that it absolutely requires this intermediate host. That intermediate host is the flea. Flea, when we acquire this infection, usually the flea will bite the mice and they'll transmit, and the mice ingest those fleas. When we come into play as an accidental but definitive host are when we ingest these fleas ourselves. So the fleas will have the larval form, this is the side in the fleas, we ingest them mature into adulthood in our intestines. We pass these eggs to the stool. The Hymenopus eggs are distinctive for these organisms, so we can identify them definitively that way. We can also release those gravid proglottids and identify as well. But the cycle continues that way. We release them in the stool, and larval fleas will feed on that stool and continue to transmit that infection that way. Because there's no risk of hyperinfection, you need an intermediate host. These tend to be milder infections than Hymenopus nana. If you do have a heavier burden, you might get symptoms like nausea, abdominal discomfort, like normal tapeworm symptoms. Okay, and the last one today, Diploidium caninum. Is this stuff making sense to you guys when we're talking through the life cycle? More or less, like definitive host, intermediate host? Okay. Okay, good. It is tricky to kind of understand this, that's why I'm going over it in a little bit more detail here. But this is my favorite one. It is, we see a lot of this in our cats and dogs. It's very common. This is one that Bella had, my cat. What happens here is an infectious cycle between fleas and normal cats and dogs. Indoor, outdoor pets, they're not on flea treatments, then they can get this disease. What do cats and dogs do when they have fleas? How do they take care of it? Yeah, they chew on those fleas and they crush them in their mouths and they swallow them open. If the fleas they swallow have the larvae inside of them that has definitive hosts, those two eggs can mature in their egg sets. So normally that continues, they pass those eggs along, and then those flea larvae feed on those infected cells that are passed from the animals, and as they mature into adult fleas, they'll have the larvae in them, ready to continue the transmission cycle. So if animals get it through eating fleas, how on earth would people get this infection? Make out with your dog. Okay. If you were to make out with your dog, you get some saliva from the animal, but no flea, right? So you can make out with an infected animal, but you're not gonna get it unless you get that flea in your mouth. The flea is what has the larvae, not the saliva from the animal. But you're thinking, what usually happens, this is why this also tends to affect kids more than adults, because kids are playing with these dogs, and kids jump from the dogs into the kid's mouth, and the kids will swallow those fleas. The fleas will get swallowed down, and the larvae that are carried by them will exit its stomach and then develop into adult tapeworms in your guts. They're very similar enough to dogs and cats, I guess, our GI tract, where these worms can actually survive through adulthood in us. But there we go, accidental hosts are capable of transmitting an infection, right? Because we can shed it in our stool. So not dead end host in this scenario. We can contribute to this infectious cycle. Good news is, in terms of symptoms, this is not a severe host tapeworm infection like you were the definitive host. You're just facing the same abdominal signs and symptoms that you normally would. Okay, but again, like if you have pruritus and these clots are migrating out of your GI tract, that's not a good thing to have. So people usually seek treatment when they have those symptoms. Let that one ride. In terms of diagnosis, what we pass in our stool is a little bit different. We have these distinctive egg packets that are unlike any of the other eggs that we've talked about. They'll stay in those egg packets until they get out in the environment, and then they break down so the food larvae can eat them. But that's a nice diagnostic feature, particularly young canine. That's it for the organisms. Just a couple of overview slides here before we call it a day. We've got teeny eggs, indistinguishable A. This could be solium, this could be saginata. Sizes are important. We have micrometers or our microscopes to measure these and know the characteristics. With B, hopefully you saw the operculum when we were talking about Diphyllobatum latum. But you can see once your eye is trained for looking for these opercula, you get pretty good at seeing that thin little lid that will pop open for Diphyllobatum. I know this Nina, that's relatively small, big egg. I know this Denuta, and then lastly those egg packets that are distinctive for Dipodium canine. On an exam, I may throw in pictures of these, but as with every other exam, if I throw in a picture, I really don't mean for it to hurt you, only benefit you. If you happen to remember nothing else about Dipodium canine other than the fact that you have egg packets, super. You know, it's just a bonus thing. Okay, quick case study. I think we still have time, right? You guys want to have a read through real quick with this case study, and we'll talk through it. Give you a few minutes to read through this. Okay, what were some things that you guys pulled out of that vignette? We'll jump straight to the diagnosis. What are some details that would be important as clinicians for you to pull out? Calcifications, take calcifications on imaging and the brain. What about the presentation, like the how did he present? What were the symptoms? Seizure? Yeah. otherwise he starts having seizures. His age, he's not very old, he's not very young, he's 30 years old. What about where he is from? Country of origin? Mexico. So not every country has the same food inspection program standards. Mexico has actually been stepping up, I think, their food inspection programs, but we do see a higher incidence of neurocystic psychosis coming from countries in Latin America. So that's interesting, that's notable. In terms of sequencing, I wouldn't put in a ton of information about, CDC and these lumbar punctures, but these are abnormal. I put the reference range for the neutrophils and lymphocytes and monocytes at the bottom there, but there's something going on when you did a lumbar puncture. He's got a lot of lymphocytes, so there's probably an infection he's dealing with. When they asked for the rule outs, what were they suspecting? What tests did they do? So, yeah, the PD, the purified protein derivative test, was negative. He hadn't been exposed to TB. Anything else that they wanted to rule out? HIV is actually negative as well. So most of these questions on the subsequent slide we've answered, but this is kind of classic for neurocystic psychosis, some neurological symptoms. Do some inducible studies. We see in this case a bunch of punctate calcifications on his brain. So we probably acquired this a long time ago, and it's just getting to the point. Maybe those larvae have died. Maybe he's getting an inflammatory response. Maybe that's why all of a sudden now he's having seizures and things like that. In terms of the infective stage for acquiring cysticercosis, whether it's muscle or neurocysticercosis, what was the infectious stage of this parasite? Was the egg? Yeah. So this is fecal oral route, and the infective stage is the egg. If he had intestinal twiniasis, the infective stage would have been larvae. Exactly. Okay. Good. Yeah. Okay. One more quick case study, and then one more slide after that, and you guys are done. Sorry. I'm quick look through this. This is classic, and let me know what you think. Alright, before we shout an answer, what is kind of giving you a hint from history yet was steering your mind towards a particular diagnosis, a differential diagnosis? Well, fish. Okay, so we have an association there. Anything else? Yeah, I can hardly pronounce it. Megaloblastic anemia. Yes, that's, I don't know the pathology there, so maybe you probably have a better idea, but that's the indication that you have some anemia going on, some low steroid levels. Symptoms? What are her symptoms? Some abdominal pain, anorexia, and she's drinking soda. So there's a geographic hint there as well. All of those hints kind of come together, and at least you start suspecting that she has diphthalitis, an infection with the fish tapeworm. There's a picture of that gefilte fish, and those fish balls that are cooking in this pot there. So how would you make that diagnosis if somebody has diphthalitis? I'll give you a hint. If I told you were the definitive host, that means that the adult's premature, right? So, are you going to see anything in that person's stool specimen on the OMP? Yes. What are you going to see? You're going to see the eggs, and you're going to see probiotics potentially, but those eggs will be percolated like bees, right? Delicatiosis will have percolated eggs. So that's distinctive in the probiotics themselves. So you make that diagnosis based on an OMP exam primarily, and those symptoms of the clinical picture. And then lastly, this is just for funsies because I had students asking about sushi-related parasitic diseases. A couple of these are tick-borne infections. A couple of these, Saki's simplex fish, but there's like just there's a bunch of other cultures who like their fish to be raw as well. Carpaccio di Succo, that is I think like a perch in Italy, pickled anchovies, tartare maison in French, ceviche, marinated herring. A bunch of cultures will have raw fish in their cuisine. So be aware that all of those could potentially be sources for parasitic diseases if they're not. In the US, we will see with sushi. We do flash freeze our fish, so we have very, very low incidence of these parasitic infections here. Sushi chefs, for some reason, they're very shy about talking about that, like they're ashamed of it, but flash freezing fish is a very good idea to kill those parasites. Okay, I'll see you guys tomorrow.